In last lecture, I have discussed the introduction part of fungi and the classification of fungi. Today, we will see the next point of our syllabus, that is the morphology of fungi. In that, first, the morphology of fungi and the difference between the yeast and mold. I have seen the difference between the mold and yeast. Today, we will see what is yeast. So, these are large, that is having the size 5 to 8 micron. Then these yeast are single cell organisms that are rarely form a filament. Yes, what is yeast? These are large, having size 5 to 8 micron. Then these are single cell organisms that are rarely form a filament. Then the most of the yeasts which are reproduced by the process of asexual process of budding. Yes, the yeast, these are reproduced by a sexual process of budding. Then on culture they form a smooth and creamy colonies. For example, Saccharomyces cerevisiae and Cryptococcus neoformans. Yes, for example, Saccharomyces cerevisiae and Cryptococcus neoforms. Then next, dimorphic fungi. The dimorphic fungi are those that exist as a two different morphological form at two different environmental setting. Yes, what is dimorphic fungi? These are the fungi or dimorphic fungi, those which exist as two different morphological forms at two different environmental settings. Then they characteristically grows at a mold in their natural habitat and in vitro at room temperature and yeast and as a yeast in culture, usually at 37 degrees Celsius. For example, Histoplasma capsulatum, then Blastomyces dermatidis. These are the few examples of dimorphic fungi. Then yeast-like fungi. Next, that is yeast-like fungi. Yes, in some yeast-like fungi or the yeast-like Candida albicans, the bud remain attached to mother cell and elongates. Yes, the yeast-like Candida albicans, the bud remain attached to other mother cells and it elongates, and which is followed by repeated budding. Yes, which is followed by what? That is repeated budding and forming a chain of elongated cell known as pseudohyphae. Yes, this is known as pseudohyphae. Then this can be differentiated from the hyphae because they have a constriction at the septa, are also present at the branching point. Yes, I am repeating here, this can be differentiated from the hyphae because, yes, what is the reason? Because they have constriction at the septa and also are present at the branching point. Then on soil media, the moist, creamy colored colonies can be reproduced. So this is the classification or the morphology of fungi. Next point, that is growth requirement of fungi. That first temperature, then pH, then oxygen, water, nutrients and light. Yes, growth requirements of fungi: first temperature, then second pH, third oxygen, next water, then nutrients and last, that is water. We will see one by one. First, that is temperature. The optimum temperature required for fungi, that is, it is in the range of 25 to 30 degrees Celsius with lower and upper limit of 10 degrees Celsius and 40 degrees Celsius. Yes, I am repeating. The optimum temperature required for growth of fungi having range, that is 25 to 30 degrees Celsius with upper and lower limit of about 10 degrees Celsius and 40 degrees Celsius. The certain thermophilic forms having the optima above the 40 degrees Celsius, and some may grow at high as the upper for 50 degrees Celsius in composite habitat. Then in another hand, there are some psychrophilic fungi that are capable of growing the, yes, some saprophytic fungi that are capable of growing the below freezing point of water. Then temperature affects, yes, what is the effect of temperature? This temperature affect the lag time. Then it also affect on specific growth rate and yield in quite different ways for each fungus. Yes, what is the effect of temperature? It affect on growth rate, lag time, then specific growth rate and yield in quite different ways for each fungus. Then higher low temperature may causes the fungi to enter the dormancy by producing the some sort of pore or the specialized structures that ensures the escape and the survival during the extreme conditions. Yes, however, the high or low temperature it may causes what it causes. It causes the fungi to enter the dormancy by producing some sort of pores or the specialized structures that ensures the escape and the survival during the extreme conditions. The next factor that is pH. The pH optimum that favors the growth of different type of fungi are vary greatly. Some fungi grows at best pH as level of 4 to 7. Then the determination of hydrogen environment for fungi is difficult because the change in the pH after they grow. Yes, I am repeating here. What is important for pH, or in case of pH, the determination of hydrogen environment for fungi is difficult. Because why it is difficult? Because the fungi changes the pH after they grow. Next, the fungi digest and release the metabolic products into their environments. The fungi are digest and release the metabolic products into their environments. They significantly alter the pH level in the microenvironment. They significantly alter the pH level in the microenvironment. Next factor that is oxygen. The majority the majority of the fungi require oxygen for oxidative phosphorylation in order to generate the energy. Yes, why it is required? Because the fungi are required oxygen for oxidative phosphorylation in order or to generate the energy. But only a few species of fungi are obligately fermentative and incapable of oxidative respiration. Yes, but only a few species of fungi, Chatridomycota, are obligately fermentative and incapable of oxidative respiration. So next requirement that is water. While some fungi live in water, the most species do not grow well when submerged in water due to low oxygen availability. Why? Because of the low oxygen availability. While some fungi are live in water, and most species do not grow well when submerged in water due to the low oxygen availability. Then the thin walled happy that comprise the mycela are susceptible to desiccation and require a more or less continuous source of water in order to or for their growth. Then the molds that are capable of growing at very low water activity are referred as xerophiles. Yes, what is xerophiles? It means the molds that are capable of growing at very low water activity, and it is called as xerophiles. For example, Erotium species and Walmia sebi. For yes, examples of those Erotium species and Walmia sebi. Those that are capable of growing at very high water activity, they are referred as hydrophilic. And for example, Stachybotrys and Chatomium, and also another example for hydrophilic molds, Holocladium. Then next factor that is nutrient. So the next requirement that is nutrient. The nutrient required for fungi may vary from species to species. Yes, nutrient required for fungi, growth of fungi, it may be vary from what species to species. And some are omnivorous and subsist on organic matter. Some molds may thrive well on substrate with high sugar or the high salt content, and some may prefer the simple sugar, and while others have ability to utilize the complex sugar. Next requirement, that is light. While light is not required for the somatic growth of fungi, it is or it may generally enhance the growth in few and be a necessity for the induction of asexual and the sexual reproductive structure. Yes, it is required for asexual or the sexual reproductive structure. Additionally, light may involve the orientation of spore bearing structure, and the spores of number of different type of fungi are discharged toward the light. Then UV radiation also reduce the, yes, what is the role of UV radiation? The UV radiation also reduces the viability of spores, especially in air. Overall, the light does not play as a major part in a metabolism and a growth of fungi. Yes, the light does not play a major role in a part of metabolism and also the growth of fungi. Next part, that is, so this is all about the growth requirement of fungi. What are the growth requirement? First, temperature, then pH, then oxygen, water, nutrients, and light. Next part, that is nutrition of fungi.